Howdy, this is lab report 4, Metabolic Test Lab. OF glucose medium, TSIA, carbohydrate broth tubes. Group 2, Ana Martinez and
And Hannah Garcia, Microbiology, Fall 2024.
Introduction. The purpose of this lab is to learn how to perform biochemical tests on different types of bacteria using different types of media. Based on the results of the lab, we should be able to identify if the bacteria can metabolize the media they were incubated with. Significance and background information. The significance of biochemical tests using different media such as the oxidative fermentative glucose, triple sugar, iron, agar, and carbohydrate broth media is to be able to identify bacteria species based on their metabolic capabilities to understand their ecological roles and pathogenicity. The materials and methods for the oxidative fermentative glucose medium. These are the materials. Start by inoculating the medium by taking a single bacteria colony with a sterilized inoculating needle. Then stop the medium to about 1/4 inch of the bottom of the tube. Repeat these steps with the rest of the tubes. Label one of the tubes as the closed tube and use a disposable dropper to cover the medium with 1 to 2 milliliters of mineral oil. Label the second tube as open and do not modify. Replace the tube cap loosely, not screwed tight. Incubate at optimal temperature for the bacteria for a couple of days, then observe bacterial growth and color changes. The materials and methods for the carbohydrate broth tubes. These are the materials. Start by inoculating the medium by taking a single bacteria colony with a sterilized inoculating loop. Replace the tube cap loosely, not screwed tight. Incubate at optimal temperature for the bacteria for a couple of days. Observe color changes and possible bubble formation. The materials and methods for triple sugar iron agar, TSIA. These are the materials, and you're going to start by inoculating the medium by taking a single bacteria colony with a sterilized inoculating needle. Then stop the TSI tube through the center of the medium to the bottom of the tube and then streaking the surface of the agar slant. Replace the tube cap loosely, not screw tight. Incubate at optimal temperature for the bacteria for a couple of days. Observe bacterial growth and color changes. The results and observations. The following slides are the results and our observations of the incubated bacteria in the test tube media. The pictures are our results and the observations based on what we recorded on our lab notebooks.
Results and observations. Oxidative fermentative glucose medium. For Pseudomonas aeruginosa, the tubes, both open and closed, were green, remaining their original color of a neutral pH. Bacteria did not break down glucose through either fermentative or oxidative pathways. The bacteria were non-saccharolytic, which means that they cannot break down carbohydrates as well. For Escherichia coli, both the open and closed tubes were yellow, which indicated acid production. Bacteria was able to break down glucose through both fermentative and oxidative pathways, which means that they are facultative anaerobes or can perform both anaerobic and aerobic pathways. Alkaligenes faecalis was green for both the open and closed tubes, remaining the original color of a neutral pH. They were non-saccharolytic, and the bacteria did not break down glucose through either fermentative or oxidative pathways. For the other Alkaligenes faecalis, the open tube was green and the closed tube was slightly light green, but they still remained a neutral pH. The bacteria, The bacteria did not break down glucose through either fermentative or oxidative pathways, so they were also non-saccharolytic. For unknown 102, both the open and closed tubes were green, remaining their original color of a neutral pH. Once again, they were non-saccharolytic and the bacteria did not break down glucose through either fermentative or oxidative pathways. However, unknown 202 was yellow for both the open and closed tubes, indicating acid production. Bacteria was able to break down the glucose through both fermentative and oxidative pathways, indicating that it is a facultative anaerobe, which can perform both anaerobic and aerobic pathways. Triple sugar iron agar, TSIA. For alkaligenes faecalis, there was a red slant of alkaline and yellow bottom for acid, indicating that this bacteria is a facultative anaerobe, saccharolytic, and only uses glucose, not sucrose or lactose. Escherichia coli also had a red slant and a yellow bottom, also indicating that it is a facultative anaerobe, saccharolytic, and only uses glucose, not sucrose or lactose. Citrobacter freundii was yellow for acid, indicating that it is a facultative anaerobe, saccharolytic, and uses glucose, lactose, and or sucrose. Salmonella typhimurium had a red slant and a yellow bottom, indicating that it is a facultative anaerobe, saccharolytic, and only uses glucose, not sucrose or lactose. Shigella flexneri had a red slant and a yellow bottom, indicating that it is a facultative anaerobe, saccharolytic, and only uses glucose, not sucrose or lactose. Staphylococcus aureus, on the other hand, had yellow color for acid production, indicating that it's a facultative anaerobe, saccharolytic, and uses glucose, lactose, and or sucrose. Proteus mirabilis also had a yellow color to indicate acid production. It is a facultative anaerobe, saccharolytic, and uses glucose, lactose, and or sucrose. In addition, it also experienced a lot of bacterial growth. Unknown 102 had and unknowns 102 and 202 both had yellow color indicating acid production. They are facultative anaerobes which are also saccharolytic and both use glucose, lactose, and sucrose. Carbohydrate broth tubes, glucose broth. Escherichia coli had a bubble at the top, indicating gas production as a byproduct of glucose fermentation. It also changed its color to yellow, indicating acid production, which lowered the pH of the medium. So it is a positive reaction. Alkaligenes faecalis also was a positive reaction, but it had no bubble at the top, indicating no gas production. Nevertheless, it did, of course, change its color to yellow, indicating acid production, even though it should have remained a red color, meaning that the bacteria should not be able to ferment glucose. Staphylococcus aureus had a bubble at the top, indicating gas production as a byproduct of glucose fermentation, as well as a yellow color, indicating acid production, which lowered the pH of the medium. Both unknown 102 and 202 had a yellow color, even though 202's color was more hazy, indicating acid production. While 102 had no bubble at the top, indicating no gas production, 202 had a bubble at the top for gas production as a byproduct of glucose fermentation. Lactose broth. Escherichia coli had no bubble at the top, indicating no gas production. It also changed to a light orange color, with very little fermentation of the lactose, indicating a somewhat positive reaction. Alkaligenes faecalis, at the other hand, while also having no bubble at the top for no gas production, retained its original orange color, indicating no fermentation of lactose, a negative reaction. Unknown 102 had no bubble at the top, indicating no gas production. It also retained its original orange color, indicating no fermentation of lactose, which meant that the reaction was negative. On the other hand, unknown 202 had a small bubble at the top, which meant that some gas production was a byproduct of lactose fermentation. It also changed to a yellow hazy color for fermentation of lactose, indicating a positive reaction. Sucrose broth. Escherichia coli had no bubble at the top, indicating no gas production. It also retained its red color for no fermentation of sucrose, indicating a negative reaction, even though it should have been able to ferment sucrose. Alkyligenous faecalis, at the other hand, had a bubble at the top for gas production, but still it retained its red color for no significant fermentation of sucrose. Therefore, it was also a negative reaction. Both unknown 102 and 202, on the other hand, had a yellow color, with 202's, of course, being more hazy, both indicating fermentation of sucrose. While 102 had no bubble at the top for no gas production, unknown 202 had a very large bubble at the top, indicating a lot of gas production as a byproduct of sucrose fermentation. Conclusion. Although some bacteria should not have been able to ferment the contents of the media, such as A. faecalis in glucose broth, while others should have been able to, like E. coli in sucrose broth, this could have been due to errors such as contamination during inoculation, not enough bacteria present in the medium, etc. Overall, we were able to test bacteria based on their metabolic capabilities and observe if they were able to break down components in media. And for the OF glucose medium test, also testing for aerotolerance. Thank you.